Amino acids is the next type of macromolecule. So let's give it a few minutes here. So, composition of an amino acid. It's got a few things that make it up. Number one, it's going to have a central carbon. So you're going to have central carbon. Number two, what you're also going to have here is you're going to have something called a carboxyl group. Now, we talked about carboxyl groups when we talked about organic acids. Because carboxyl groups are the thing that we said are the coolest. So you're going to look for that COOH. And then the other things you're going to have is you're going to have a hydrogen. And this last part where it derives its name, you're going to have something called an amine or amino group. Now, an amino group is going to be a compound that contains NH3. Or I should say, it'll have nitrogen in it. So, here we go. We're going to diagram it. Now, I will tell you, this is a rather large diagram. If you look at my sheet here, right, it's got a pretty large diagram that makes it up. So, what ends up happening is amino acids, what they do is they bond to each other, and a whole bunch of amino acids join together to make a larger structure called a protein. So a bunch of amino acids makes up a protein. So the monomers are going to be the individual amino acids. And so what would that look like? Well, it would look like this. You're going to have an N initially. Off of that nitrogen, you are going to have two hydrogens coming off like so. And then you're going to have a carbon that is bonded to something called an R group. Now that R group, this thing right here, is kind of funky. It's weird. And what that is that's something called a central carbon group. And this R will change depending on which amino acid that you have. Okay? Also coming off of this is a hydrogen. And then coming off of this carbon as well is going to be another carbon with a double bonded O and an OH. And this right here is one amino acid. Okay? Now, what will happen is this. This will then bond together with another one. I got to write a little smaller here because I got to be able to fit this large drawing in. But, again, you're going to have a carbon that's going to be bonded to a nitrogen. It's going to be bonded to a hydrogen, an R group, right? Then a hydrogen, a C, an OH, and double bonded O. Now, just to give you a couple of things, right? Your hydrogen is here. You're going to have your hydrogen is here, I should say. Your amine group, your NH2 is right there. There's your central R. And your carboxyl group is this C double OH. And what ends up happening here is you end up with a bond occurring called a peptide bond. And when a peptide bond occurs between two amino acids, again, we end up making a larger thing known as a protein. What's made of protein in your body? Things like your hair, your skin, muscles, etc. So what I'm going to do is I'm going to draw this from left to right. It's going to be a large molecule. So here we go. You're going to have an H bonded to an N. Then what you'll also have bonded to that N is you're going to have still a hydrogen. So here's an NH2 group. And that NH2 group is going to be bonded to a carbon, which is bonded to an R group. And that carbon is still going to have a hydrogen coming off of it like so. Then what happens is this. You're going to have a bond. You're going to have a C double bonded O. And here's where you're going to have your water that came out. Okay? And here's why. What's going to occur here is the N of here is going to be bonded to the carbon here, because what we'll do is right here, this H and this OH right here, there's H2O, so we do a dehydration synthesis, we kick the water out. So what does that do? It leaves this left over. So we end up with a nitrogen bonded to a hydrogen, that's that right there. We have a carbon bonded to an R. Still going to have a hydrogen coming off of it. Then you're going to have another carbon coming off of that, a double bonded O, an OH, right? And there you go. And this whole molecule right here is known as one protein. And we could build lots of proteins so we can make lots of skin, fingernails, and hair, etc., etc. So these are your individual monomers. So that's why I have a plus right here. One amino acid, another amino acid making this big thing called a protein. So, what are our monomers and what are our polymers? We just went over that, but let's make sure we're thorough and that we're covering it, in a good fashion. So, the monomers here. The monomers are the individual amino acids. Therefore, the polymer is the protein that is made. Now, the bonding, I talked about this earlier, the bonding that occurs to make a protein from amino acids is called peptide bonds. So we have a peptide bond. How does that happen? It happens via a dehydration synthesis. And just like we've been talking over and over again, I feel kind of like a broken record here. When we make a bond, we do a dehydration synthesis. But if we want to break it, what are we going to do? We're going to do hydrolysis. So to make it, we did a dehydration synthesis, right? If we wanted to break a protein down into its individual amino acids, we do a hydrolysis reaction. Now, with proteins, the best example that I can think of, I've given you a couple of them, okay? But these are large, complex, polymers of amino acids. And one of the ones that I would like us to focus on is something called an enzyme, okay? And what an enzyme is it's a protein that acts as a catalyst in a chemical reaction. So in a chemical, and then we'll write RXN, because that's what I always write for reaction. It's the chemist coming out of me. What a catalyst means, this word, it means it speeds up chemical reactions. Speeds up reactions. And how does it do that? What it does is it takes something called the activation energy, which is necessary to make a reaction occur, and it lowers that activation energy so that the reaction can happen sooner. Okay? Now, enzymes, so enzyme diagram, how they work is like this. I have keys, right? And my keys, are specific to a particular place. So, for example, this key would get you into my classroom, right? But this key wouldn't get me into the gym. This key wouldn't get me into, the art room. This key is specific to a particular door and lock, right? And so enzymes are the same way. And so what we end up making is something called an enzyme substrate complex. Okay? Enzyme substrate complex. And what happens in an enzyme substrate complex is it obeys something that is commonly referred to as the lock and key hypothesis. Now, specifically, what is the lock and key hypothesis? It's this. Let's say we have an enzyme, okay? And that enzyme is shaped like Pac-Man. So we have an enzyme that looks like this. And there are two possible substrates that it comes across. One which is shaped like a triangle, and another that is shaped like, like a star. Okay, so these are two possible substrate combinations. Well, you can see here that this particular substrate would fit very easily right in here, right? And so what ends up happening is this is the appropriate substrate to fit in that enzyme so that we could make something called an enzyme substrate complex. But this star wouldn't fit. So this is something like we talked about, amylase versus cellulose. Your body has two things in it, right? Or has the ability to break down starches, but not cellulose due to the presence of enzymes called amylase. And that's because it's specific to a substrate, starch. Okay, they link up. They're a lock and a specific key. So, proteins, what are we going to use them for? Okay, so uses and biological applications, just to finish up our notes. Okay. Well, what are we going to use them for? The big thing that we're going to use proteins for is we're going to, make new proteins. What does that mean? Well, we talked about it earlier, right? We said, sometimes we might want to make skin. Sometimes we might want to make hair. We might want to make fingernails, etc., etc. So anything in your body that's composed and made up of protein would be governed by amino acids. And then the other thing is this thing called enzymes. And again, enzymes facilitate and speed up chemical reactions. And that's amino acids.